Welcome back to Fun Facts Online. Today we are stepping into the dark to see the light. I want you to start by imagining a room that is absolutely pitch black. You cannot see your hand in front of your face. But suddenly, suspended in the air right in front of you, a constellation of stars ignites. It is not random. The lights flicker and pulse in a rhythmic dance. One area flares up, then the light travels across the darkness to ignite another cluster. It is beautiful, but it is not a planetarium. You are looking at the interior of a living brain, and every single flash of light represents a thought, a memory being recalled, or a command to move a muscle. For decades, this vision of a self-illuminated brain was pure science fiction. If we wanted to see the brain work, we had to force light into it. We had to drill holes, insert fiber optic cables, and blast delicate neural tissue with lasers to make it glow. We treated the brain like a passive object that needed a flashlight to be seen. But as we close out the year 2025, a quiet revolution has fundamentally altered our relationship with the mind. We have entered the era of autoluminescent mapping. We no longer need to shine light on the brain, because scientists have effectively taught the brain to shine for us. This is a massive shift from fluorescence, which is reflecting light, to bioluminescence, which is creating light. Today we are going to do a deep dive into this technology, how it works, and why it is going to unlock the secrets of sleep, social behavior, and memory in ways that were physically impossible just a few years ago. To understand the gravity of this shift, we have to look at the old way of doing things. When I say the old way, I mean the methods that dominated neuroscience from the early 2000s up until very recently. The gold standard was fluorescence. You have probably seen those glowing green videos of neurons firing. Those are created using proteins like CaBLAM. The way it works is that scientists genetically engineer a neuron to express a protein that glows green when calcium floods the cell, which happens when a neuron fires. But here is the catch. Those proteins are not lamps. They are more like glow-in-the-dark stickers. They only work if you charge them with a bright external light source. This created a massive engineering bottleneck that has plagued neuroscience for 20 years. To get that charging light deep into the brain, researchers had to do some pretty invasive things. First, they had to implant hardware. We are talking about cementing optical fibers, or heavy, miniaturized microscopes, onto the skulls of mice. Second, and this is the big one, the animal was often physically leashed to a laser bank. Imagine trying to go about your daily life, interacting with friends, or trying to sleep, while you have a fiber optic cable glued to your head and a laser blasting blue light into your skull. It restricts movement. It restricts natural behavior. And third, there is the issue of phototoxicity. Blasting biological tissue with high-intensity blue laser light is damaging. It heats the tissue. It alters the physiology of the cells. Eventually, it kills the cells you are trying to study. Plus, just like a poster left in the sun, fluorescent proteins fade over time. This is called photobleaching. You might get 20 minutes of good data before the signal vanishes. That makes it impossible to study long processes, like sleep cycles or memory consolidation, which take hours or days. So the result of the last 20 years is that we learned a lot about the brains of mice that were stressed, head fixed, and enduring laser blasts. We knew far less about the brains of animals behaving naturally in the dark. This is where autoluminescent mapping flips the script. It draws inspiration from nature's own engineers. Think about fireflies, deep-sea jellyfish, or glowing fungi. These organisms do not need a laser to glow. They perform a chemical reaction inside their bodies that releases photons. This is bioluminescence. The concept is elegant. If we can borrow the genetic code that makes a firefly glow, which involves an enzyme called luciferase, and splice it into a neuron, the neuron becomes its own lamp. Now, for years, this idea failed in practice. Natural bioluminescence was simply too dim. A single neuron firing produced so few photons that no camera could detect it through the skull. The signal was lost in the noise. It was like trying to see a candle flicker from a mile away in a thick fog. But that changed with the development of hyper-bright, synthetic bioluminescent indicators, culminating in a breakthrough known as CaBLAM. That stands for Calcium Bioluminescence Activity Monitor. Developed through a collaboration between the Bioluminescence Hub at Brown University and researchers at UC San Diego, CaBLAM is the superstar of this field. It is a genetically encoded sensor that solves the dimness problem. It is a molecular machine composed of a specifically engineered luciferase enzyme coupled with a calcium sensor. I want to break down exactly how this molecular machine works because it is fascinating. Think of the brain being supplied with a fuel. In this case, it is a safe, non-toxic molecule called a luciferin, specifically a synthetic analog like furamazine. This is the gas in the tank. When a neuron is at rest, the CaBLAM molecule is in the off position. The enzyme is physically distorted, so it cannot burn the fuel. But when the neuron fires, calcium ions rush into the cell. These ions bind to the sensor, causing the molecule to snap into a new shape. This shape change opens the enzyme's active site, allowing it to oxidize the luciferin fuel. That reaction releases energy in the form of bright photons. The result is a neuron that flashes brightly the moment it processes information. But the brightness is only half the story. The true genius of autoluminescent mapping lies in a concept called signal-to-noise ratio. This is the most critical part to understand. In traditional fluorescence, the laser method, the brain is flooded with external light. This creates a lot of noise. You get autofluorescence from blood vessels, the skull, and other tissue that glows faintly when hit by a laser. Trying to see a specific neuron firing in that environment is like trying to see a flashlight beam outside on a sunny day. The background is just too bright. Bioluminescence, on the other hand, works in the dark. The brain itself does not naturally emit light. It is a pitch-black canvas. Therefore, when a CaBLAM neuron fires, even if the light is technically fainter than a laser-excited fluorophore, it stands out with infinite contrast. It is like seeing a single candle in a sealed cave. The background noise is effectively zero. This zero background property allows researchers to do two incredible things. First, they can see deeper. Photons generated deep in the brain, in emotional centers like the amygdala, can travel out through the tissue without being drowned out by the scattering of excitation lasers. Second, and this is the game changer, they can remove the hardware. Because the light is coming from the brain, sensitive cameras placed outside the skull can detect it. There is no need to insert a fiber optic cable into the brain tissue itself. This removal of the laser leash has opened up entirely new categories of neuroscience research. Let's look at three specific areas where this is changing what we know about the mind. First, the social brain. Have you ever tried to study social interaction between two mice when both are tethered to fiber optic cables? It is a tangling nightmare. The wires get crossed, the animals are stressed, and they do not behave normally. Autoluminescence allows for wire-free recording. Mice can chase, groom, fight, and mate naturally, while cameras overhead track the neural firing associated with these complex social behaviors. We are finally seeing the social brain as it actually functions, not as it functions when restricted by laboratory hardware. Second is the sleeping brain. Sleep is a long process. It cycles over hours. Traditional fluorescence bleaches out too quickly to capture a full night's sleep, and the heat from lasers can disturb the animal's rest. If you heat up brain tissue, you change how the neurons fire. Bioluminescence is chemically powered and generates no heat. Researchers can now monitor brain activity continuously for hours or even days, tracking how memories are consolidated during REM sleep without ever waking the subject. Third is developmental neuroscience. Because autoluminescence is non-invasive and requires no implants, it can be used on developing brains. We can track how neural circuits form in young animals over weeks, watching the lights of the brain organize themselves into mature patterns. This was previously impossible because implanting hardware in a growing skull causes severe damage and disrupts the very development you are trying to study. And the innovation does not stop at tracking electrical activity. There is a parallel breakthrough from MIT known as BLUSH, or bioluminescence imaging using hemodynamics. This combines bioluminescence with MRI technology. In this technique, blood vessels are engineered to dilate in response to bioluminescent light. This effectively turns the brain's own vasculature into a massive 3D camera. When neurons light up, the nearby blood vessels react, and that reaction is captured by an MRI scanner. This allows for deep brain mapping of the entire organ at once, bridging the gap between the single-cell precision of CaBLAM and the whole brain scale of fMRI. So what does the future hold? We are standing at the threshold of a new era. Autoluminescent mapping is moving us toward a read-write relationship with the brain that is purely biological, rather than mechanical. The non-invasive nature of this technology holds immense promise for medicine. While we cannot yet genetically engineer human brains to glow, this technology is accelerating drug discovery right now. We can test epilepsy drugs on glowing mice to see in real time how a drug suppresses a seizure storm across the entire cortex, rather than just at a single electrode site. Perhaps most futuristically, the bioluminescence hub is exploring intercellular communication via light. This is the concept of the Internet of Brains on a cellular level. If one neuron can emit light via bioluminescence, and a neighboring neuron can be engineered to be activated by light using optogenetics, we can create synthetic circuits where neurons talk to each other using photons instead of neurotransmitters. This could allow us to rewire damaged brain circuits after a stroke, using light as the bridge to bypass broken connections. For centuries, the skull was a black box. To see inside, we had to break it open. Autoluminescent mapping has turned that black box into a glass house. By metabolizing the very energy of life into light, we have given the brain a voice that we can see. We have moved from the harsh glare of the laser to the soft, informative glow of the biological star. As we look at these maps of glowing activity, we are not just seeing data. We are watching the physical manifestation of a thought, burning bright in the darkness of the mind. The brain is no longer a dark planet waiting for our flashlight. It is, finally, a star. That is all for today's episode of Fun Facts Online. Thanks for listening to this deep dive into the future of neuroscience.